get started. Today, it's my pleasure to introduce Dr. Anita Mahadevan-Jansen. Many of you know her quite well from many years in the photonics world. She is a professor at Vanderbilt Photonics Center. She investigates applications of optical spectroscopies and imaging for disease diagnosis, so a lot of translational as well as basic development. She had her master's degree and undergrad degree from Mumbai University and master's degrees in biomedical engineering from the University of Texas and PhD from the University of Texas. And she's been at Vanderbilt since 1996. She holds the Orrin Ingram Professor of Biomedical Engineering position at Vanderbilt University, and she's also appointed in the Department of Neurology, Neurosurgery, and Photolaryngology. She has a very impressive history of research and publications, and really a pleasure to have you come and talk with us today.
Thank you. Thank you all. I was last here about 4.5 years ago. When I was visiting, because my daughter was exploring UCI as a place for undergraduate school. At that time, because I was going to all these universities and I knew people there, I was also going around giving presentations since it gave me an opportunity to do that anyway. And one of your students had asked me next time I came to town if I would be willing to present, and I said, sure, why not? So when Hannah asked me for a title of my talk, I told her something, and then I was without internet and I couldn't remember what I told her. So, I mean, I knew what I wanted to talk about. I knew what I sent her for the abstract, but I couldn't remember the title. So I said, you know what, I'm going to start by giving you an overview of the Vanderbilt Biophotonics Center that I started and run, and then go into my own research and give you some vignettes of a couple of different projects of mine. So that's what you'll hear. I do talk with my hands and I do like to walk around, so bear with me. That's just something, that's the educator in me that can't help but walk up and down the hallway, and I'll go back there and I'll wave my hands around. So promise not to kick anybody's pizzas out of the way. I usually start with this slide, which is sort of the mission and vision for the Vanderbilt Biophotonics Center, just formed in 2015, although I should say that this building was the Vanderbilt Free Electron Laser Center from those days of the military free electron laser program. For those of you who've been here for a long time, you know what I'm talking about, since Beckman is part of that. And the laser used to sit 2 floors down. The floor 1 below this ground was where all the labs were, and then this floor is where the offices are and a couple of labs, and up here are 2 operating room suites, and where the laser was taken up there to actually use for medical neurosurgery applications. When the program got shut down in 2009, I started working hard. At that time, I was just a recently tenured faculty member, and I was looking at the senior faculty at Vanderbilt to do something with the space and the synergy that was already there across medical center and various parts of the university. But I had to wait till I was a full professor before I started walking the pavement, so to speak, to get Vanderbilt to form the center. And so we finally became a center in 2015. The mission then of the center, as a bunch of faculty getting together saw it, is really about seeking to foster the next generation of biophotonics researchers. So education is sort of the key part of the mission of this center. And then the research side is really to push the boundaries of what light can do, both for fundamental discovery as well as clinical translation. Vanderbilt came out with a t-shirt that said impact, innovation, and some of the I words. So I started thinking about it, and I already had translation in there. So I came up with 2 more T words, and I made it transstitutional and transformative. So that's where those 3 words come from for the next t-shirt that we make, by the way. The center collaborates with a whole bunch of various institutes and centers within Vanderbilt, and also a lot of institutions, and there are many here that I just stopped finding logos and sticking them in there because it's just getting packed. But the idea is to interact both internally and externally with a variety of different places. Geographically, Vanderbilt as a private university is a very compact campus, and yet we sit dead center, okay? And so you have the primate center on one side, all the medical stuff here, College of Education here, engineering more or less here. We have a VA, a Veterans Hospital, psychiatric, cancer center, eye institute. So you see that as a biophotonic center, it's beautifully placed right in the middle of all of this. The biggest thing that, one of the things that we've been pushing for, I've been pushing for, is that the center is really more of a collaborative research base rather than a user facility. So nobody comes in there expecting to use an instrument. The idea is they come in there to work with us to solve a problem and develop a project. That's sort of the fundamental tenet of what the Vanderbilt Biophotonics Center bases itself on. And so we have faculty somewhere on the order of 40 different PIs from all over Vanderbilt University and Vanderbilt University Medical Center. That's what those two acronyms are referring to, because about 3 years ago, Vanderbilt University and Vanderbilt University Medical Center are separated into two legal institutions, financially different institutions. So we're all still on the same campus. So it's still right here, nothing's changed, except they are a separate financial entity compared to the rest of the university. But regardless, I have an appointment in both places that allows me, and as do most of the faculty members in biophotonics and biomedical engineering, and we work with various folks across campus. Both they come in, they work with folks in the center, but do their own research at various places. You're looking at roughly on the order of $32 million in research funding that, as I mentioned already, intersects with a variety of institutes and centers on the Vanderbilt campus. So one of the things we wanted to do when we started the center was really create a thematic focus or a thrust area, for want of a better word, where we excel. We show ourselves that we excel. So the three main areas that we work on, by the way, when I first started, this used to be called cancer photonics. And then we realized we do more than cancer, so it became clinical photonics. Clinical photonics, neuro photonics, and what I'll call multi-scale photonics, and I'll explain what that is in just a second. Give the three main thrust areas of what we do, but ultimately what we do is solve problems. Okay? Because we are centered so nicely in the middle of campus, people come to us wanting to solve problems, whether it's medical or biological, and then we brainstorm looking for light-based solutions, because that's what we know, to solve that problem. And so, as I mentioned, some of those things don't quite fit in those categories when we want to look at bone biology or embryo mechanics or preterm birth. They don't quite fit in those categories. But regardless, it's a good PR value, obviously, to have nice three sectors, and then in terms of recruitment, a lot of different things. The idea is, of course, to just apply light to solve problems, both in engineering, medicine, and biology, focused primarily on biomedical applications. So what's multi-scale photonics? We have faculty in electrical engineering who work on nanophotonics, which is looking at single molecule detection kinds of things, to MALDI, and a variety of different light-based approaches to look at single molecules. So there's sort of, we have a strength in nanophotonics. Something that we really started to push is what I'll call microphotonics, which is developing microscopic techniques that are not available commercially, to make it available for the biologists to make fundamental discovery. So what that means is we were able to get a couple of $1 million from the institution to bring in, for example, Eric Betzig's lattice light sheet microscope. We built it at my center, down on that second floor. This video here is the first images we got from the light sheet. Let me see if I can play that. I'm not sure how. Okay, since I don't have mouse control, is Richard around to tell me how I'm supposed to play my video? Because the video is playing here, but I'm not seeing it play there. Anyway, those are microvilli of the small intestines in the digestive system. The idea is, you can see them dynamically over time. Sorry, I don't know how to play that video with the tools I have in front of me, even though it's playing over there. It's not playing over there. Because even though yeah. Because this is not in presentation mode here, even though it's in presentation mode there. So somehow it made extended desktop or something. My apologies. So what did you do? So I know I have a couple more slides.
Split the mouse all
Oh, okay. So it is an extended desktop. Okay.
Right now it is. Yeah, that's the right side.
Okay. Got it. Thank you. So these are essentially, you can look at these cells and look at the microvilli and do dynamic imaging over multiple hours without killing the cells with lattice light sheet illumination that allows you to really extract a lot of information. In this particular case, we were interested in seeing how the changing microbiome of the gut changes the motion mechanics of the microvilli. So we also have swept sort of angular oblique incidence, planar illumination, light sheet microscopes. There are multiple acronyms that are used. You've heard SCAPE, you've probably heard SOPI. There's multiple people doing different. So this is where you use a single objective to create the planar illumination. We have one of those as well. If we have time, I'll also show you another one that we call Mantis. That's what we mean when you're looking at multi-scale photonics. It's looking at everything from the protein level up to whole organ level. Okay, so Neuro Photonics, something we pioneered in my lab, which was using infrared light to activate or modulate the nervous system, everything from single cells all the way to the human body. And so we have a whole area that's focused on that. We do have some optogenetics, bunch of activities on imaging neurostructure and function using a variety of different techniques, as well as disease detection. One of my very first projects was actually brain tumor demarcation. And then finally, the last area that I'll call clinical photonics, which is really where you're developing techniques for clinical translation. This particular area is kind of interesting in that almost all the projects in this area come from clinicians walking to us, residents or attending, saying, I'm having this problem in the OR or in the clinic. Can you help me find a light-based solution? In fact, yesterday I got an email from a pulmonologist working in conjunction with a pulmonary surgeon who came wanting to look at small granulomas in the lung and finding ways to screen and diagnose it. So is there technology out there? Yes, the first task is always go do a research and see, literature search and as we do things that nobody else is doing. That's been an important thing for me. Is like if somebody else is already working on it, they're ahead of the game. I don't want to compete necessarily. It makes sense if I have some kind of unique solutions or somebody in the center has unique solutions that has never been tried before. So most of the projects you'll hear about as I go along will be things that we sort of pioneered in our lab. So what you'll see is we have projects that are related to screening, projects that are related to diagnosis, variety of conditions, not just cancer, guidance of therapy, just detecting physiology that I've already talked about, as well as point of care diagnostics. By the way, this is like projects across the whole center, all faculty, not just mine. And so I go back. So the idea really that we try to push is what can light do? So to push the envelope of what light can do. So this is my sort of termination point about telling you about the center. As I said, about active group, about 40 PIs across both parts of campus. The goal is really highly collaborative research. So when people come to the center and have labs or equipment in the center, it's, the understanding is if the instant you put it in the center, it means you make it available for anybody on campus to be able to use it. With babysitting, don't get me wrong. You're not just going to say, here, go use it. But that's sort of the understanding. We really encourage and push students to pursue individual fellowships, not just training grants opportunities. So we have a variety. Almost all our students are self-funded. Junior faculty mentoring, both on the medical side as well as the engineering and science side, is a big part of the center, and funding from all over the place. And you can see more from our website as well. Okay, with that, I want to switch gears and really talk about my own research or my own research philosophy as well. As I said, what I do really is problem-based research, which means that, whether that application happens to be detecting cancer, finding a plaque, understanding physiology, guiding surgery, my goal on all my students is understand the clinical problem that they are trying to address, understand the relevant anatomy and physiology, identify the need, and then identify the technique or technology that best allows you to solve that problem. Okay, so there's no one technique. I use all kinds of techniques. You know, you can start through the acronyms of all the different techniques. The only technique we haven't used is photoacoustics, but other than that, you know, pretty much any of the techniques are open game as long as we need to solve that problem. So my own research has now 4 main areas. I always talked about having 3 main areas, but now has 4 main areas. Optical guidance of surgery was one of my first areas of project. The brain, as I mentioned, was the very first project I've worked on. And since then, there's a whole bunch of various. The idea here is using real-time intraoperative guidance using light. Optical detection, where I'm using various techniques on looking at things along the lines of cancers, but also bone quality and fracture risk, inflammatory bowel disease. In fact, we have a lot of work now looking at inflammation-based conditions, preterm birth, and I'll give you some examples, or some sort of various ways of looking and understanding both pathology and physiology of tissues. Infrared neural modulation, I've already mentioned, and then this translation of novel microscopies. So my goal, I hope, will depend on time and how fast and how much I talk, is to give you a snippet of in each area if I can. I might not get past preterm birth. So I want to tell you a little bit about parathyroid, which is sort of what I talked about before. If you were there then, you probably heard it. I want to give you an update in that case. Preterm birth, because that's an area that I'm really excited about. Something about modulating cells, not just in vivo. They'll give you a snippet. And then if I get a chance, I'll show you the mantis microscope in case you're wondering what that acronym stands for. All right, so let's start with the parathyroid. So the parathyroid gland is a small gland the size of a grain of rice that sits right behind the thyroid in your neck. And it actually is the only organ in your body that regulates calcium. It secretes the parathyroid hormone and sends a signaling in order to tell the bones to produce more calcium. The other organs of interest, if you look here, are going to be the thyroid, of course, because that's where the 4 parathyroid glands are sitting right there, but also lymph, fat, muscles, and nerves. The clinical problem arises from the thyroid and parathyroid surgeries that occur, or the field of endocrine surgery as it's called, where there are roughly about a couple of 100,000 people a year who go through these surgeries, whether it's for a variety of conditions in the thyroid or a variety of conditions in the parathyroid. But more the problem, the challenge is to identify these organs, whether they are normal or not. So this is anatomical guidance. You just need to find the gland. Doesn't matter whether it's diseased or not. Find the gland and tell the surgeon where it is so that they can do their surgery, whether it's thyroidectomy or parathyroidectomy, as in whether they're trying to remove the thyroid gland or trying to remove the parathyroid gland. The biggest problem here is inadvertent removal or injury of the parathyroid because they are so small that can occur during these procedures. In fact, if anybody's ever had thyroid surgery or knows anybody who have, in the fine print, it explicitly states that you could have, your parathyroid could get injured, you could have lifelong problems with calcium as a result of that procedure. Okay, that's something you always sign off on because it's a very well-known side effect. Now you have 4 of them, so you injure one, you give your body time, your body can accommodate, but sometimes it cannot. It depends on what's going on during the procedure. It is by far the most sort of single largest cause for litigation, malpractice litigation after endocrine surgery is injury to the parathyroid gland. If you go on blogs, you will often see a statement that says, can I sue my surgeon because I have all these problems with calcium now? There's a lot of statements like that. Anyway, so as I said, the clinical challenge to us, and this project, by the way, came to us from a 3rd-year surgical resident who had watched me present about my brain tumor work, knocked on my door and said, hey, this is a problem we're having, and I'm seeing this is a problem whether it's an experienced surgeon or me for finding this gland. Do you think there's a light-based solution to look at this? I told her, well, somebody's got to have done that before. So let's go do the literature search and come back. And she came back the very next day and she said, couldn't find anything for the gland. There's a couple of papers looking at parathyroid cancer, 2, actually, exactly 2. But other than that, there didn't seem to be anything. So I said, okay. So we went to the lab and got some tissues, okay, and looked at UV visible fluorescence. Didn't really see anything different. Looked at diffuse reflectance. Too much fat in the way, all you saw was fat reflectance, couldn't really see anything. Did OCT, didn't really see anything. Finally did Raman spectroscopy because that was okay. If it's that sensitive, maybe you need to do that, and got that. Every time we looked at the parathyroid gland, the signal saturated. Every time we looked at the thyroid, you know, if anybody here do Raman spectroscopy? Okay, that's Raman. Those little bumps are Raman for those of you who don't know that. Okay, the rest is background signal. Every Raman person knows there's always background signal that you work with. So something of this background signal saturates or is high. But although it took us a while, you know, we at first glance are like, something is wrong. Signal detector, there's stray light coming in from somewhere. You know, you think little LEDs are somehow getting into your detector, and you block off little tapes on every little light source in the room, and we still kept saturating. In fact, we went ahead and said, okay, let's go into patients, and took our Raman system in first 30 patients, and still saw the signal saturating. It took me about 30 patients to say, wait a minute, maybe this has nothing to do with Raman at all, and maybe this background signal is actually a fluorescence signal or a luminescence signal that's actually causing, and that's what led us then to build a lab-based system that we then went and tested over the next few years in about 137 patients, okay, going through thyroid or parathyroid surgery, measured something on the order of 264 glands, and have histology only from diseased parathyroid glands, because normal parathyroid glands don't come out, and showed that you consistently get a really strong parathyroid signal, whereas every thyroid has some, everything else not so much, with about 97% accuracy. I wanted to show you this, and that's sort of a threshold. So we've done a whole bunch of different kinds of diseases, okay? And then you just want to look at what the detection rate was. And you'll see it's up in the 90s, except for this one case called secondary hyperparathyroidism. In that particular case, 50% of the glands glow, if you want to call that high fluorescence glow, and 50% didn't. But in this particular case, all 4 parathyroid glands come out anyway as part of the surgery procedure. So if you remove that, you're at about 99% accuracy in 137 patients. If you've worked in optics long enough, you know it's a very rare thing to get 99% accuracy in patients. So that was our lab-based system where OR lights was a problem, so you had to do it in the dark. You need to do post-processing of the data, and we used a sterilizable probe that was actually quite expensive. And we licensed that idea anyway to a company actually out of Santa Barbara that was called Anisys Instruments. They build microscopes, nano IR microscopes, that then spun out a medical device company called AI Biomed, leading to a clinical prototype device that looks like that. It's called PTI, parathyroid identification, can be used with the room lights on, has an audible indicator, built-in analysis, and a foot pedal. All you need really is a bar that says whether your signal is above 1.2 or not compared to the thyroid, okay? And has a disposable probe to go with it for a marketing model. So we tested that and consistently got, you know, results that were 99% sensitive, a little bit lower in terms of specificity, submitted this data. By the way, all of this was done where the surgeon was still blinded for all of these procedures. So the surgeon didn't get to see the results. You know, they got the answer after the fact, but we submitted that to the FDA and finally just got FDA approval in November of 2019. So now this is now a product. This is Jiju Thomas, who was the postdoc who was responsible for all the clinical studies, which is why he's on there, and we published that multiple times. So that's now out there at various surgeons' hands in order to be the first adopters to take that forward. Unlike in that I did not just sign a license agreement and told them, okay, take care of it, go develop your product. It's something I've continued to have a relationship, not a financial relationship, but a grant-based relationship to build this technology to move it forward. Because in my mind, this is probably one of those few things that's going to go quickly through market. I don't know if I'll invent another thing that will quite do that. I hope so, but I want to make sure this is there in every surgeon's hand. Just recently organized an endocrine surgery conference where we had endocrine surgeons from around the world come, and this is clearly going, near-infrared autofluorescence is clearly going to change how endocrine surgery is going to work going forward. because there's no other technology right now. There's nothing in competition when we invented this to do this. Oops, let me go back. But you know, this is a spectroscopy device, as in I have a little probe that goes around touching it to tell them whether it's suspicious for parathyroid or not. What the surgeon wants is something where they can see, and that is a parathyroid, and that is a thyroid, and the signal is quite strong. This is excised, but it's quite strong, okay? So they want spatial context, right? And the signal is so strong that you can actually use any ICG-based camera. It's in the same wavelength range, and it'll work. And people started doing this. In fact, multiple companies exist that have ICG systems that have, in fact, tested it for the parathyroid application without licensing the technology and selling it, okay? Somebody told me, don't get mad, take it as a compliment because somebody's emulating and copying you and duplicating your work, and I've tried to do that. Can you tell it's not quite working? But these are all systems that do qualitative imaging. They just look at room, you know, you turn the lights off because they work ICG, and they're all meant for ICG fluorescence, which is very bright. And they work as in they just, what you visually see. They're not sensitive enough quite. So we've started to say, what can we do that's better than all of those things, right? And still keep our edge in the market, so to speak. And so one of the techniques, you know, in traditional surgical, you know, when you have an imaging technology, it's usually on a screen and the surgeon is doing surgery and they're looking out there to do this. I said, well, that's not, you know, plus it's not quantitative. So we wanted to do quantitative fluorescence imaging, but also do it where the surgeon does not have to lift their head from the surgical field. So while I was at a conference, I realized, hey, wait, we have these projectors. So we're just going to project what we image right back to the surgical field. Okay, and there are microprojectors. If you've gone and looked on Amazon, you can find microprojectors that are about that big, right? About this, the size of my hand, or even smaller. And so that's what we did. We went to Amazon and bought a projector, okay, and put a system together. First generation had this big one. This one is about that big. Next generation is already much, much smaller. And so we built a system that has a CMOS-based camera system that then projects, collects the information from the camera and projects it right back onto the surgical field. It looks like that. That's a real case. That lets you see the parathyroid gland is there, and when it processes it in real time, I can actually have it show up on the surgical field. We called it OTIS, or Overlay Tissue Imaging System. Okay, and that was OTIS 1, and we already have OTIS 2 and OTIS 3 actually right now. So these are two OR actually in the intraoperative case of a diseased parathyroid gland and a normal parathyroid gland. That's why this one is small and that one is as big as it is. And we were able to do this with 100% success. I think that's my next slide. Yeah, so the other thing we discovered when we started doing this was the first question the surgeon asked was like, this is great, but can you also tell me when the parathyroid blood supply is cut off? Okay, so people have been using ICG for exactly that, because that's what it's approved for, right? To look at blood supply. And what we found, well, we're like, well, actually Bernard had published a bunch of work on laser speckle imaging for burn and for flap, and Andy Dunn has similarly looked at blood vessels. I'm like, what's the simplest technology where we can look at blood supply? Was laser speckle imaging. So that's what we did. We developed a laser speckle imaging system so that when you have the gland, you see the speckle contrast. In this case, it's not, it's viable. In this case, it's not, it becomes lighter. And we can tell by looking at the speckle contrast value when a gland has become compromised. So now we have a dual system that will both identify the fluorescence and therefore identify the gland, as well as tell the surgeon, both label free, whether it's viable or not. There we go. So as I said, PTIs with the first adopters. Lots of imaging systems are being tested. All of them, commercial ones, require the room lights to be turned off. And so whether we can do label free without the room lights turned off. So we've built our latest prototype system that will be out there being tested. We'll go from there. In this case, we can go after 510K. By the way, this went through a de novo process. So if you know anything about patenting or, sorry, FDA approval process, the de novo people always dread because it takes forever. It took us about less than a year. because there is no predicate device. Okay. All right. So that was the first vignette I told you I'd tell you about, and we are at roughly where I thought I'd be, which is about 30 minutes into my presentation. So we'll do preterm birth, and as I said, then we'll look at the clock and we'll go from there. Okay. So under optical detection, so my PhD work was on cervical dysplasia using Raman spectroscopy. At that time, very few people, if almost nobody, had done in vivo Raman spectroscopy inside the body. They'd done it on the skin, okay, on the breast through the skin, but nobody had actually done it inside the body. And so that was my PhD work, build the very first in vivo Raman probe to look at cervical dysplasia. I continued to do that work. At first I did not, because I thought my PhD advisor would continue to work on it, but she actually did not. So a couple of years later, I continued to work on looking at using Raman spectroscopy for cervical dysplasia. And the reason I'm telling you, there's a motivation to this. As I said, you know, there's always a method to my madness, so just bear with me. So what we discovered was that it was great to be able to tell high-grade lesions from normal, but the technique was not great for being able to tell low-grade from normal, or metaplasia or inflammation, other conditions that exist. And well, Raman spectroscopy is a biochemical fingerprint. If there's any technique that would work, it may be weak, but it should work. So I asked the question, why was it so poor? Turned out that Raman spectroscopy is very sensitive to hormonal changes. So I could predict if somebody was in the pre-ovulate, before ovulation, or after ovulation, using just looking at normal patients. Okay, I could tell. So that means I started thinking, okay, what's the biggest change a body goes through, a woman's body or the cervix goes through over the course of the life? Pregnancy. So I started looking at pregnancy, and I'll tell you what I discovered, right? 15 million preterm births occur each year, okay? And it's a direct cause of a third of infant deaths, preterm birth. What is preterm birth, by the way, is defined as anything below 39 weeks, but when we look at babies like this, these are like sub 30 weeks, usually 27 weeks or even less. And what is most amazing even today is that in 50% of the cases, we have no idea why preterm birth occurred. So you're talking about these little babies that are the size of your hand that are born, and every day you can keep the baby inside the mother's body, it increases their survival rate by something on the order of 15%. Every day, okay? So, okay, so the two things met, right? The problem and what seemed like the solution, which is being able to look at hormonal changes. And so the idea was, well, the issue though, before we could even get to in terms of detecting preterm birth, was not much was even known about this problem of what happens, right? I told you 50% of the time we don't even know what's going on. So pregnancy, it's well known that the cervix remodels during pregnancy. It goes from being a thick, you know, organ to really thin one that dilates and is elastic. In fact, cervical change precedes uterine changes by many, many weeks, mainly considered of extracellular matrix changes. Proteins are known to change till you get to dilation phase, till the ripening phase. Other things are changing. Clearly, there's a lot of things going on, and by looking at the cervix, we will just learn more about the pregnancy process, also what happens in preterm birth. So the thought process, but working with a neonatology collaborator of mine, Dr. Jeff Reese, was, can we look at cervical remodeling, the biochemistry of cervical remodeling, to learn more about preterm birth? My hypothesis was, and still is, that there has to be biochemical signaling changes that has to occur well before mechanical changes happen. Like before that labor the signals can go to tell the uterus to start contracting, for the cervix to start dilating, the mechanical changes, there have to be biochemical triggers ahead of time. So if we can detect them early, then we can potentially do interventions to prevent or minimize delay, let's say, a preterm birth. So that was sort of the fundamental idea. And clearly I had already shown that Raman spectroscopy can look at the cervix in vivo, in real time. We can look at the cervix over time, over the course of the pregnancy. We can look at biochemical and structural effects, and we can look at all the things that are known to be changing, whatever little bit is known, while the cervix remodels. So with that in mind, we got IRB approval to do a study at the Women's Health Center at Vanderbilt, where we enrolled ultimately about 68 patients. We did monthly measurements in the first through the first 2 trimesters, and then weekly measurements over the last trimester, just whenever they came in, and that's usually when they came in to make measurements. And we took a Raman system, by the way, that's really the Raman system, and there's nothing else in there. Laser source, spectrograph, CCD, a probe, control with a laptop, and just touch the cervix on the 4 phases of the clock, 12 o'clock, 3 o'clock, 6 o'clock, 9 o'clock on the ectocervix, made 3 to 5 measurements in multiple locations in the cervix, just gently placing. It did require a speculum exam because we need to be able to see the cervix to make that measurement. That was the only extra step that we had to ask these patients to agree to. And we measured on the 68 patients, and we roughly got, results all over the place, but you can see there are spectral changes. Colors, meaning as you go more pink, you're further along, okay? Including postpartum, and we have 68 patients worth of this data. Now you have multiple points in the cervix over time for multiple patients, right? So you have to develop statistical analysis tools in order to be able to extract information. And today's buzzword, you've got to use machine learning, and if you want AI and all those buzzwords to get to the data. We did multivariate statistical analysis, which is the same thing, okay? That's a pet peeve of mine, by the way. You use the terms for grant proposal writing, but the reality of what you're doing is multivariate statistics. Now, the question is whether you let the machine make your decisions or you make the decisions. That's the only difference between the two. We did some analysis in order to extract out what was overall the general trend across all 68 patients over time, okay? Over gestation time, and what changes as you get further along. Things we see that people had never really ever recorded were that there's increased amount of blood signals, okay, as you go closer to term. There is decrease in collagen, which has been seen. There are other changes. You'll see some more blood-related signatures, some actin, collagen, essentially extracellular matrix changes that occur, but people had never really noticed that blood changes. Now, if you ask the clinician, they go, oh yeah, the cervix always looks more pink when they're further along. Of course it looks more pink, there's more blood, essentially, right? So we have now signals that show that factor. And then when you extract out various components, you can really start to make some trajectories of how the collagen signals are starting to go down because the collagen gets diluted and disorganized over the course of pregnancy. The blood signals go up. You actually see some lipid changes that go dramatically up at labor and postpartum. So we just saw a whole bunch of things, especially related to cholesterol or lipid signal and actin signals that dramatically increased postpartum. Which is another finding. Most people had not really looked at the postpartum cervix. Most people just tend to look at, mostly in animal models, but look at the during parturition process. And when you look at the wound healing literature, it made perfect sense. Essentially what you were picking up was the wound healing response the cervix was going through going through the trauma of delivery. So we looked at other variables, first-time pregnant mothers versus women who had had multiple babies, and you really see huge changes in their vasculature in the first-time moms, but there's not as much changes. In fact, in almost every category, you don't see as much changes in the spectral signatures that you see for nulliparous versus multiparous parent moms. When you look at body mass index, it's been shown that women with a high body mass index, it takes them longer, their whole pregnancy cycle, they tend to have delayed delivery, it takes them longer to go through the process of pregnancy, and that also makes sense because it seems like everything just takes a little bit longer. The collagen is much, much slower to remodel and change compared to somebody who's at term at a normal body mass index. So those are some other findings that we published at the American Journal of Obstetrics and Gynecology. The other thing we did, we also looked at, all of this work was done by my graduate students, of course, The other thing we did was we decided we also wanted to see what happens during labor. So we looked at patients who are being induced, because that's easy, they're predictable, we know when to be there, and follow them over the course of the labor they go through after they've been induced. They check the cervix every 4 hours, so we can do that. And we saw huge changes in the lipids signals in these women going through labor, and there seems to be a trigger point at which the labor is actually successful. And so we could correlate these ripple peaks with hours until delivery almost directly. Suspicion is likely due to prostaglandin surge, but I don't have the answer as to is that what it is, because we're in the process of looking at that. As I said, the one requirement in recruiting these patients was the fact that we needed to do a speculum exam. Speculum exams are uncomfortable, especially for a pregnant woman who's worried about so many other things. So we started thinking about how can we do this, especially as now we're ready to start recruiting patients who are high-risk patients, patients at risk of preterm birth. We want to not, if we can, introduce the speculum. And so we designed a probe where we would be able to introduce a Raman probe with a camera in there so we can see where we're going during bimanual exam, digital exam as they're called. And so we built the probe, this is what it looks like, and we've tested it. I don't have the tested data, but we've tested it, and we're in the process now of recruiting essentially patients, high-risk patients in our fetal maternal clinic. And so in a few months, in at least 9 months, probably more like 15, 16 months, given the number of patients we want to recruit, we should hopefully have data to show you how the cervical remodeling changes in patients at risk at preterm birth. I will tell you that we have done sister in parallel with human pregnancy, which does take 9 months when it's normal. We've also looked at mice who take 19 days, so you can do a lot with 19-day pregnancies, and we've studied both animals who have been induced to have preterm birth versus normal, and we can predict. We've most recently been able to predict when they will go into labor within 2 hours in an animal, of when, within a 24-hour period. So I think we're getting there in terms of being able to our vision of what we want to do. So as I said, we've successfully used Raman to monitor cervical remodeling in normal pregnant women. Term pregnant women is probably more accurate. Collagen signals significantly decrease. Blood signals significantly increase. Postpartum, we see effects consistent with wound healing. There's lipid changes associated with labor, and we now have a speculum-free probe that allows us to do this. So here's a study where clinical translation is the goal, but it's going to be longer term before we get there, compared to the parathyroid one, where the solved problem, the solution was much, much shorter in time frame. Here we need to first understand what was going on and understand pregnancy before we get to the goal or the target of identifying patients at risk of preterm birth. How are we doing on time? What is our goal of when we need to finish by? 1. Should I stop so you can ask me questions? Or we can go ahead and ask me questions and we can see where we are and I can continue. Because this is complete shift of gear. I think someone wants to ask a question, so go ahead, please. Yes. Yes, that is what's going on in our mouse models. And we can also do some other things like introduce DII and actually excise the cervix at various time points in the mouse cervix and look. I'll have the answer in a few weeks. Uh-huh.
What kind of challenge is that using modern?
No, absolutely. The nice thing about children, especially really, really small children, is actually, that's a whole nother discussion as a pediatrician, is that babies are wonderful optical models because scattering is really low. Okay, that's my scientist hat, not my mom hat, okay? You gotta remember which hat I'm wearing. They're great models for light because, so we have other projects where we're looking at things like PDA and sepsis and so on in babies' light. But in this case, we're looking at adult mom cervix And we can do quite a bit with depending on wavelength choice. So one of the projects we have is using laser speckle imaging to look at the blood vessels, so we can say whether we have increased vasculature, increase in small blood vessels, or actually just blood volume changing. We'll be able to do that in pregnant women. Yes. Yes, so that part, I didn't talk about that part of the research. That part of the research is going to India and sub-Saharan Africa to actually do the testing of the cervical cancer part. Yes. Yes, absolutely. That's correct. So that's where the cervical, in this country, it became too high, you know, it didn't make sense to continue cervical dysplasia, but that's what we are in the process of doing, is getting approvals in India to be able to look at cervical cancer there. Yes. We know we can. That's not the problem. It's can we make the device cheap enough to be able to do it there? That's the problem. Uh-huh. Uh-huh. Yep. Yep. It's huge. Absolutely. Yeah. Yes. One of the projects in our global health is actually detecting gastric cancer, for example, in Honduras and other LMIC countries. For the same reason, we have Vanderbilt physicians who go there on a regular basis and residents, and we can get, you know, 200 patients in less than a week because of that. So, yes. Yep. Yes, exactly, in the same visit. That's the goal, is the see and treat idea. Yeah, that's right.
Yeah, that's really interesting. Do you know what the source of the autofluorescence is in parathyroid? And what other organ systems it's used in?
Yes and yes, sort of, I guess I should say. For the slides that I eliminated was what's the fluorophore slides, because I was going into giving snippets of various projects. But what I can tell you is that we don't, it's hard when you have no idea when the molecule is, because you can only eliminate stuff, and we've eliminated about 15 different things. It's not calcium sensing receptors, it's not vitamin D, it's not precursor, we thought it was chromogranin, it's not chromogranin. What we have arrived at, it's a small molecule that has a mass of around 15 kilodaltons or lower, has fluorescence lifetimes on the order of 1 nanoseconds or lower, and lower, I guess I should say. There are two things, there are two lifetimes. And it's very, very, very resilient. It requires 24 hours of buffer in order to extract out that fluorescence, but normally it takes 45 minutes. So, yeah, exactly, yes. And the other question, what else? There's a few other endocrine, yeah. But there are a few other organs that do have fluorescence, and we can talk about them.
So if you're moving, if the surgeons are moving with thyroid, has anyone tried to encapsulate the parathyroid tissue and put it in another compartment of the body?
Yes, so that's pretty standard. That's why they want to know whether it's vascularized, because if they can take it out, they can mince it up and put it in your arm, and it'll still do the frame. That's parathyroid is a miraculous organ that will do that, actually, which is why they want to know whether it's viable or not. There's one more question. It's found in the cytoplasm of the chief cells. But nobody specializes in parathyroid histology, really. They just want to know if it's the parathyroid for all purposes. So that's how far we've gotten in terms of anatomically, microanatomically where it is. So we're at 10 minutes to 1. Okay. Okay. All right. So here's another video. If y'all see the mouse, let me know, please. No, go back. I'm not seeing the mouse. Oh, there it is. Yeah, I gotta go there. Hold on. Okay, so when you take infrared light delivered through a fiber and put it on a nerve, for every pulse, you get one twitch. Okay, the amount of light you put in is very small, well below the ablation threshold, since Boston is there, I can say that. Well below the, and with safety ratios in the order of 2 or 3 times in terms of doing this. And as you move it spatially along the nerve, you'll see different, that's what this is telling you, different muscles. You go from this way to that way to things, only that muscle twitching. So you can selectively decide which nerve fascicle you're actually stimulating and therefore causing a response. So that's the fundamental basis of infrared neural stimulation, okay? So it's the direct deposition of transient deposition of infrared, pulse infrared energy onto the nerve to evoke an action potential. The mechanism in terms of physics is thermal gradient based. And there's been modeling work that's shown that you're essentially effectively changing the capacitance of the membrane, okay? The more what's happening from that energy change to cause currents, ion channel flows and current is something that is part of the thing that's currently funded and it's ongoing. We have some answers and I hopefully will give you some of them. You can do it contact free, it's damage free, it's artifact free, and gives you a high spatial specificity. So we've actually been able to show this from everything, and I put my pointer down. In everything from the peripheral nervous system in vivo, you just saw that, central nervous system in the brain, both in vivo and ex vivo. With collaborations at Northwestern, we've been able to show in the cochlea for cochlear implant purposes. There's videos online if you want to look. I forgot to put it on here, that you can also use it to pace the heart in a quail embryo heart, but still in a heart. Okay. So there's lots of parts to this. I'm not going to talk about all the parts, but what I thought I would tell you, one, we started with the FEL. Okay, we went to a Homium YAG, and now have a diode laser that's the size of a shoebox. Okay, all through military money. This technology got licensed by a company named Aculite. That's a laser company that made infrared countermeasures for the Apache helicopter. That company got bought over by Lockheed Martin, who insisted they wanted the medical part of Aculite, and then 2 years decided they didn't want it. So I now have the technology back, okay? But we still nevertheless managed to develop a laser device, optical stimulator, if you will, as part of this technology for animal research. When we do it in vivo in the brain, even if you don't understand all of this stuff, just look at this. And when you do repeated, the mechanisms are slightly different. We can use infrared light to stimulate and inhibit the brain, as well as stimulate the brain based on parameters and where we place it. Whether we are inhibiting the excitatory neurons or exciting the inhibitory neurons, I do not know. Okay? This is research. It's part of the fun of what we do. We just see stuff and then we got to figure out what's going on, right? So, but we've been able to do this with no damage, in fact. This was done in the rat somatosensory cortex and visual cortex. We've done this in humans. Patients, cerebral palsy patients who were undergoing dorsal rhizotomy, where they go in and use electrical methods to identify a few nerves and see what it innervates, and then they cut a few. Usually kind of intelligently random way, okay? I didn't say that quite right, but you know what I'm trying to say. But then they cut it, okay? That's all they do, they cut. So when they cut, it also means I can take a piece of it and see if I cause any damage with light. So we were able to do this both for efficacy as well as for histology purposes. And what's nice is we can do side-by-side comparison of electrical versus optical. And electrically, when you stimulate one particular nerve, you see multiple things in the left leg started activating. You take the same nerve and use optically, only one thing activated. These are all various electrodes of all the different muscles in the body. This was a 7-year-old kid, because cerebral palsy is mostly about doing surgery on kids. And we were able to really be more targeted as to which nerve rootlets they should cut. because you know exactly what muscle it's innervating. That was sort of the idea. We did it about in 10 patients, and we showed that we can really get damage, you know, very safe, non-histological, and remember, nerves will actually rejuvenate and get better, recover after some period of time. This is all acute studies. We still had safety thresholds of about 0.9 joules per centimeter square or lower to be able to do this successfully in patients. All right, we've done it in an awake monkey where we've actually made, so this was, you know, you saw jerks, right? Like one pulse made a jerk, which is great, but not really useful. If you think about man-machine interface, you want them to be able to do something coordinated. We've been able to make a sea slug actually go and feed even if they didn't want to by using light. In this case, we made a monkey that was trained, a non-human primate, not a monkey, a non-human primate, look somewhere else. They were supposed to be fixated on a spot, and then you use light on their visual cortex and force them to look elsewhere. Okay, and then we looked at, stuck them in an MRI to see, did it under MRI, and we could really see what was going on with the brain. Anyway, mechanism-wise, I'm going to skip over that slide. One of the things we found when we looked at the central nervous system, and I killed this pointer, is that you saw a fast signal as well as a slow signal. And in electrophysiology, you can tell that what that means is one of them is a neuronal component and one of them is an astrocytic component. Which one comes first? And therefore, the whole question of can you actually use light to activate astrocytes became an important question for neuroscientists. And so we did that. I'm going to skip that. Okay. Now, these are both videos that actually show what light does to astrocytes. And I'm going to see if I can at least get one of them to play. There it is. It doesn't matter. You can see when the laser turns on, there it turns on, you activate the astrocyte, and then it goes away and goes back to its resting state. Okay, and you can do this repeatedly multiple times. This is a single pulse effect. This is a multi-pulse effect. You can see it also communicates to other, even though we're only eliminating one astrocyte. And you can see essentially a field effect. What we've derived, and you don't need to read all of this, essentially is that while TRPV channels are important for this process, what we discovered that really the trigger is aquaporin 4, as in water channels, that take calcium, extracellular calcium, into the cells. That's what seems to be important, which in turn has an effect on the TRP channels. So that's the biology side, the transduction process that we're still trying to extract out. But this is sort of the most recent paper with these results that is currently submitted that we hope to be able to do. Because what this does is if you can modulate astrocytes selectively, that opens up a whole bunch of ideas around immune suppressants and things like that in the brain. If I can activate microglia or astrocytes selectively, then I can send those cells when there's, you know, damage, modulate them and send them where I want to potentially, or activate them to do something protective, and especially neurodegenerative and functional disorders kinds of things. Okay. With that, that's sort of, I gave you a little bit of a snapshot of the nerve stimulation part of it. And I will end with letting you see what a monster microscope looks like. This is called the multimodal, nonlinear, and thermal imaging system. That's what MANTIS stands for. So it can do 2-photon, SHG, CARS, SRS, and thermal, all with 1 microsecond resolution and 1 micrometer spatial resolution or less. Okay, we built that with Thorlab's help. That's the kind of images you can get, which is composite of all of these various things. And if you zoom in, you can see myelin sheets. Okay, when you look at this, you can see individual nerves. This is a nerve, and you can see the myelin sheath of the nerve and the nodes around the air. Sort of, just to give you sort of the idea of the level of things you can do. All right, so with that, we can do this in vivo and ex vivo, by the way. Lots of people to thank, okay, for all the different projects I talked about, all the funding sources, including like student grants and stuff like that. Thank you for your attention.
We have time for about 2 questions.
I want to see if any. Yes.
So you said the activation of the astrocytes and neurocytes is due to a thermal gradient. Can you go into the mechanism a little bit more? Because could it not also be vibration or from other sources?
Sure. We are using energies very well. We've done things like you can use a hydrophone to cause the same kind of thermoelastic expansion where you're only looking at the pressure effects and you don't see the kind of activation, consistent activation. But the level, you know, given how much we produce, we don't produce a lot for these wavelengths. So we've done that. We've looked at just whether it's a pure temperature effect, and it's not. You really need the gradient in order to, you know, just changing the temperature by X number of degrees. If you've made the nerve cold, doesn't cause it. You have to get the delta. So we've done that. We know we've done wavelength dependent responses, and we know we just seem to follow water absorption and penetration depths. There's no miracle molecule that's somehow causing this. That's why water ends up coming up as being one of the big contenders for figuring out the mechanism. And if we can, in fact, trigger aquaporin 4 channels, there's a lot of really cool implications on that. If you follow the opioid research at all, you know, aquaporin 4 shows up there big time.
Is there a possibility that you could use that technology to better understand what others are learning?
That's one of the things we're working with the Air Force Research Lab on, because they're interested in looking at vagal nerve stimulation and what that does to learning, and we hope to be able to learn more on that area. It's tricky when you do vagal nerve because so many other things are also modulated when you go near the vagal nerve, but it's one of the things we're working with the Air Force, some autonomous stuff like your heart rate. So we're working on that. I think he had a question.
Well, with your in vitro studies using infrared light, how sensitive can you get? Are you down to a single nerve resolution? Or
We can. We can do single cell. Mm-hmm. We've done single nerve fascicles as well. Yes.
Thank you.
Thank you.